Chloroplasts, like the ones shown here, are responsible for photosynthesis in plants, and they get their color from chlorophyll, a special pigment that absorbs light from the sun, which is located in the thylakoids inside of the chloroplast. Here's a basic look at the two stages of photosynthesis. On the left, you have the light-dependent reaction, which takes place in the thylakoids. On the right, you have the Calvin cycle, which is also known as the light-independent reaction, which takes place in the stroma. Basically, you see the H2O being split and giving off O2 as a byproduct, and the CO2 being incorporated into the Calvin cycle and creating glucose as a product. This diagram shows how a photosystem harvests light. When a photon strikes a pigment molecule in a light harvesting complex, the energy is passed from molecule to molecule until it reaches the reaction center complex. Here, an excited electron from a special pair of chlorophyll A molecules is transferred to the primary electron acceptor. The diagram represents linear electron flow, where light from the sun drives the synthesis of ATP and NADPH by energizing the two photosystems embedded in the thylakoid membranes of the chloroplasts. It's known as the linear electron flow because electrons flow from photosystem 2 to photosystem 1 through various proteins in the primary acceptors. A protein found in photosystem 2 is plastocyanin, and a protein found in photosystem 1 is ferredoxin. Essentially, these help move the electrons either through electron transport chains to help create the byproducts of this light reaction. Cyclic electron flow takes place only in photosystem 1, where photoexcited electrons are occasionally shunted back from ferredoxin to chlorophyll via the cytochrome complex and plastocyanin. This reaction only creates ATP and no NADPH. This diagram shows a comparison of chemiosis in mitochondria and the chloroplast. In both kinds of organelles, electron transport chains pump protons across a membrane from a region of low hydrogen concentration to one of high hydrogen concentration. The protons then diffuse back across the membrane through ATP synthase, which drives the creation of new ATP. This diagram shows a more detailed version of the light reaction, as up top you see the linear electron flow, and on the bottom you see chemiosis, where an ADP is being attached to a phosphate, creating ATP. The products of the linear electron flow and this chemiosis will both go to power the Calvin cycle with the NADPH and the ATP. This diagram details the Calvin cycle, where a 5-carbon sugar, RUBP, is attached to a molecule of CO2 and catalyzed by the enzyme Rubisco. The product of this is 2 molecules of 3-phosphoglycerate. This step in the cycle is known as carbon fixation. The next step is known as reduction, where each of the 3-phosphoglycerate receives an additional phosphate group from an ATP, becoming 1,3-biphosphate glycerate. The NADPH then reduce this compound, which makes it become G3P. One of these G3 molecules will be given off as a product, while the rest will be turned back into RUBP in a step called regeneration. This diagram shows the C4 pathway. The structure and biochemical function of the leaves of C4 plants are an evolutionary adaptation to hot, dry climates. This adaptation maintains a CO2 concentration in the bundle sheath that favors photosynthesis over photorespiration. This diagram shows some of the difference between the adaptations of C4 and CAM plants. C4 takes place in different types of cells with the mesophyll in the bundle sheath, and then CAM just takes place in the same cell but at different times of day, so during the night and during the day. Diagram is a review of photosynthesis. with water, carbon dioxide, and light as your reactants, and oxygen and a sugar, generally glucose, as your products. This diagram is an overview of cellular respiration with glycolysis taking place in the cytosol, the citric acid cycle and electron transport chain taking place in the mitochondria. Glycolysis and the citric acid cycle both undergo substrate level phosphorylation, while the electron transport chain undergoes oxidative phosphorylation. Substrate level phosphorylation is where some ATP is made by direct transfer of a phosphate group from an organic substrate to ADP by an enzyme. Glycolysis can be divided into two phases: energy investment and energy payoff. During the energy investment phase, the cell actually spends ATP, but the investment pays off because more ATP is created during the energy payoff phase. The ATP is produced by substrate level phosphorylation, and the NAD plus is reduced to NADH by electrons released from the oxidation of glucose. The net energy yield from glycolysis per glucose molecule is 2 ATP plus 2 NADH. Glycolysis also gives off 2-pyruvate, which help to fuel the Krebs cycle. So essentially the pyruvate is transformed into acetyl CoA as the NAD plus becomes NADH, CO2 is released, and the coenzyme of A is added. This is a basic look at the citric acid cycle. So the acetyl CoA gives off 2 CO2, creates 3 NADH, 1 ATP, and 1 FADH2 per spin around the cycle. So one spin equates to 1 pyruvate, and 2 pyruvates are created from glycolysis. So for one glucose molecule, it'll go around the citric acid cycle twice. This diagram gives you an in-depth look at the electron transport chain, which consists of 3 complexes of integral membrane proteins: NADH dehydrogenase, complex 1, cytochrome C reductase, complex 3, and cytochrome C oxidase, complex 4. Also, it contains 2 freely diffusible molecules, ubiquinone, which is also known as coenzyme Q, and cytochrome C. This diagram gives a close-up look at the ATP synthase, where hydrogen ions in the intermembrane space travel across the ATP synthase into the mitochondrial matrix. The hydrogen ions are attracted by the electronegative oxygen located inside the matrix, and as the hydrogen ions pump through this rotor, ADP is attached to a phosphate and creates ATP. This is known as oxidative phosphorylation. This diagram shows the coupling of the electron transport chain with chemosis. So you see that oxygen acts as like the final electron acceptor. So it shows the electrons passing through the different complexes, pumping hydrogen across the gradient, and then the hydrogen coming back into the ATP synthase, creating the ATP. This whole process is all oxidative phosphorylation, and it occurs, the hydrogen is pumped from the intermembrane space back into the mitochondrial matrix. So here's a review of cellular respiration. So our anaerobic respiration without oxygen is the stage of glycolysis, where our glucose is split into 2 molecules of G3P, which is then oxidized to produce pyruvate and 2 ATP, as well as 2 NADH, which are electron carriers, which will be used later in the electron transport chain. The 2-pyruvate from glycolysis, we now are entering aerobic respiration, so requiring oxygen, are turned into acetyl CoA, which go into the citric acid cycle, which produces another 6 NADH, 2 FADH, another electron carrier, and 2 ATP. Now we're going to take these electron carriers and pump them through the electron transport chain, and through chemosis and our ATP synthase, we'll create another 32 to 34 ATP. So per glucose molecule, we're producing anywhere from 36 to 38 ATP.
Filipino got the sauce.